Good morning, sir.
See, I want to take two classes at a stretch. If it's possible, shall we take it today itself? Matlab, I will not take entire 2 hours. Rather, I'll take 1.5 hour or so. I'll try to complete both.
Yes, sir.
Yeah, because
Yes,
last
sir.
two classes I could not take because of several issues. Network मतलब मेरा बिल्कुल बैठ गया था, so I was getting out randomly, and that's the reason why I could not take the class. So if possible, we can complete it today itself.
Sure, sir. No problem.
Won't be an issue, sir.
Yeah. Because we will be able to complete chapter 4, and then we can leisurely do the chapter 5 part and assignment part as well if there's any. Okay. So I'll start in a while. Let others join. Now we have 59. Let the numbers go till 65 or something, then we can start. Meanwhile, I'll just open the presentation and keep it ready. Yes, Ankit, tell me. Ankit, you had raised your hand.
Sorry, sir, it's a bit my mistake.
Oh, okay, fine. After medicine, we have actually three chapters. Chapter number 3 is of bioenergetics, where we have metabolism, enzymes, and photosynthesis part. Okay, so metabolism already we have briefed earlier. Midseम से पहले ही हमने brief कर दिया था, so we haven't मतलब did it after the midseam examination. We did it before only. and enzyme and photosynthesis हमने recently खत्म किया है. So chapter 4 में we are supposed to have biosensors, molecular machines and bioremediation. So timing we are keeping bioremediation aside. Okay, अगर time permit करेंगे तो we will do bioremediation also, otherwise we won't do it. And so in chapter 4 we will be, for now we will be studying molecular machines, biosensors. And chapter 5 we have 3 topics again. We have nervous system, immune system, cell signaling. So immune system हमने फिर उसको loop में लगा दिया. So we will be completing nervous system, cell signaling. Once everything is done, we will discuss among ourselves. मतलब हम faculty discuss करेंगे कि सब classes में topics खत्म हुआ है या नहीं हुआ है. If time permits, then we will complete the topics. Otherwise, we will go ahead with the cut syllabus. Okay, so syllabus में एक या दो topic को हम हटा रहे हैं. From chapter 4, it's bioremediation. From chapter 5, it's immune system. So these two we will not be doing as of now.
Okay, sir.
I think it's better we start. So we will begin with biosensors. So we complete this topic. After we complete this topic, we'll take a short break of 5 minutes or 6 minutes, and then we'll complete the other topic also. Okay. Yeah. So is my slide visible to everyone?
Yes, sir.
Yes, sir.
Okay. So we are going to begin with biosensors. So what are biosensors basically? Usually sensors हम कहाँ पे use करते हैं? We use the sensation for many of the mechanical aspects or many of the technical aspects. Okay, where you should be getting an input and the input has to get recognized, and based on the recognized signal only we are able to perform a task. Okay, I mean in case of any technical field or in case of any so-called mechanical aspects, we do that. Okay, the same thing also works for the biological system also. So we depend on the stimuli, right? As soon as we receive any stimuli, we tend to behave, we tend to work. So how does this stimuli basically initiate a reaction? So when the stimuli is nothing but it's like a external factor or an internal factor. This factor comes and binds to our biological molecule. Usually we're all made up of chemicals, right? So any of the biochemical, when it interacts with another component, they bring about a change in the physical property or a chemical property, and we should be able to identify what changes are happening. Okay, and if we are able to recognize that, then we can use it as a sensor. Okay, we can use it as a sensor. So what is this component that actually can do this job? We call that component as transducer. Okay, so transducer is one of the kind of a devices which is able to so-called recognize a set of factor. Like it can be a heat energy, it can be mechanical energy, it can be any sort of physical energy, or it can be chemical energy, thermal energy. Okay, jitna bhi energy based concepts hai, most of these energy conversions usually happen using this particular device called as transducer. Okay, so what we do is that we take a transducer and we try to fix it to our biochemical element. This biochemical element, when we are going to give it to a analyte, the analyte will be binding to the bioelement, and these two will be bringing out a physico-chemical changes, and that's what we are able to see, and that's what we are able to interpret, saying that this phenomenon has happened, which means that so-and-so levels are higher or so-and-so levels are lower. It's basically for detection of any sort of a biological event or a non-biological event as well. Okay, but mandatorily we are going to use two components here. One is our biochemical element, which is a which is going to act as your sensing element, and then the conversion element, so-called transducers. Okay, so this converts one format of energy into electrical format of energy. Okay, usually we are actually looking at the transducer, which can give you an output in the form of electrical signals, because we want to see the readings. Okay, we want to see a digital display. Right, so we usually get a recording or we usually get a reading of any device in this panel. So ये reading पाने के लिए the signal should be electrical, the output should be electrical. Okay, so यहाँ पे input, it can be any format of energy. So how does this biosensor came into existence? It came into existence since the invention of electrode called as Clark's electrode. Okay, so this fellow named Leland Clark, He was the one who invented Clark's electrode. So before his invention of so-called Clark's oxygen electrode, the so-called doctors, when they used to perform any surgery, they used to face a lot of difficulty in monitoring the oxygen levels in the patient. Okay, क्योंकि patient के अंदर oxygen level काफी important है. And if something is going wrong with respect to oxygen level, चाहे आपका सर्जरी ठीक से जा रहा होगा तब भी पेशेंट सर्वाइव नहीं कर पाएगा because you don't know what's happening with oxygen level. So this is like one of the most important inventions at that particular era. And after his invention, most of the doctors were able to do a safe surgery, and the success rate was too high. And this way, millions of the people around the world every year are getting saved. Okay, so this started the so-called evolution of biological sensors. At that time, at that point of time, the biological sensation used to happen in a very less context. But as of now, if you're going to look at the present scenario, we have too many devices recording too many types of events. Okay, and that's how the so-called biosensors is like one of the progressing fields in the current date. Okay, so how does the biosensor work? Visually, we have to look at analyte. It can be of analyte kya hota hai? This is something that you want to identify about. It can be sugar, it can be the pollution level, it can be a DNA, it can be a bacteria, it can be your virus, it can be anything. Okay, so when you have an unknown thing and when you want to identify what this is and how much it is present, then uska exact binding partner ko hum lete hain. That binding partner is nothing but a biochemical which tends to show interaction with this thing, and we call this as biological element. So this biological element, thermodynamics vary when it is bound to analyte, and that change in the thermodynamics are recorded by the functional element called as transducer. So this is the technical element that we are using. Okay, so we have a combination of a biological element and a technical element, and these two what we do is that we try to attach them, two of them together. We try to couple them together. So we have different types of mechanisms through which we can actually couple them. Like we have mechanical elements, and we have chemical bonding, then we have physical absorption methods. We have different types of methods. Okay, so through these methods, we are able to make them into one piece. Okay, so this is now going to act as your biological sensor. Okay, so this transducer, what it can do, it can actually convert any format of signal into electrical signal. So as you can see here, if you want electroactive material to be changes happening at electroactive level, then you have to use an electrode. If you want to look at the pH change, then you have to use a pH electrode or a semi-conducting pH electrode. Then if you want to use a heat-based changes to be recorded, then we have to use thermistors. For lights, photodetectors. For mass change, piezoelectric media. So this way, what you want to observe, what kind of change you want to observe, based on that change, you have these many versions of transducers. Okay, and what do? They give an output. They give an output in the form of signal. Oh, where did it go? I'm sorry, just give me a minute. So transducer का काम क्या होता है? It basically detects what is actually coming, what kind of signal, depending upon what kind of signal you want to record. We have different types of transducers. Okay, and we will be able to identify the incoming signal through that. Okay, so output, usually transducer का function क्या होता है? एक format of energy को दूसरे format में convert करना. Okay, एक तो आपका sensation, one is the sensing part. So it does make look at the senses. What type of changes?
Your screen is not visible.
Yeah, I'm sharing it. Meanwhile, I'm just discussing it. I think it's visible now.
Yes, sir.
Yeah. Okay. So this is so your first component, analyte, can be anything. So analyte का जो specific binding partner है, that will be your biological element. For example, let's say we have an antigen here, then it should be antibody here. For example, let's say we have a DNA here, उसको complementary DNA यहाँ पे होगा. Okay. For example, let's say there is a substrate here, then it can be an enzyme here. So depending upon what you want to look, your bio element vary. So analyte में बहुत सारे components हैं जो हम detect कर पाते हैं, and उसका जो binding partner है they also tend to vary. and these will be your biological elements. Okay, so these biological elements are fixed onto your transducers. So based on what you want to see, based on what changes you want to see, you have to use that kind of transducer, and that will be giving you an output. So if your output is weaker, in that case you can actually use these many things. मतलब आपको समझ लीजिए बैकग्राउंड का नॉइज ज्यादा आ रहा है, या समझ लीजिए आपका सिग्नल काफी वीक है, जो आउटपुट निकल रहा है वो काफी वीक है. In such cases, what you can use is that you can use an amplifier, or you can use a filter, you can use a lineager compressor. There are many other technical devices which you can employ in between to enhance or to nullify the background noises or clear the signals. Okay, so when you want to get a clear data, you can use these extra elements, and you can get the output. So output should be electrical signal every time. Remember that the output should be electrical signal every time. So these transducers has the ability to change these formats of energy into electrical outputs. So this is how your biosensor basically is established. So it works on the same principle because most of the biological phenomena have enzymes being involved. Okay, enzymes का activity काफी होता है हमारे body में, biological systems में, because as we have discussed earlier, majority of the metabolic reactions do involve enzyme activity, and hence we can simply use enzymes for many of the reactions to take part. So enzyme का function आपको पता है, it binds to substrate. Enzyme substrate will be bringing out a change in the activation energy, and this is going to give you a product. So in this conformation, we are going to do a recording. Okay, so in this particular state, we are going to do a recording, and this is what the transducer will be able to pick up, and this is where we get the signal. Okay. Now, depending upon the kind of the transducers, we have 5 different types of biosensors. The first one is a resonant biosensor. Second one is optical. Third one is thermal. Fourth one is ion sensitive. Fifth one is electrochemical. So beginning with the resonant. Resonant, usually this we will be using it for detecting the changes that are happening at the mass level. So when you have a specific mass, okay, आपका जब mass बढ़ जाएगा या घट जाएगा, the kind of the resonance that is established will be disturbed. Okay, so we will be able to recognize using an acoustic wave transducer. So the acoustic wave transducer will be looking at the wave frequencies or changes in the mass. Okay, and this automatically will be depicting the changes in the mass, and that's how we will be able to see what are the changes that are happening at the analyte level. Okay, so हम इसके लिए पहले क्या करते हैं? We use a small known mass. So with this known mass, we make it interact with this bioelement. The bioelement is usually coupled onto a piezoelectric device. So this piezoelectric so-called transducer will be able to now record the surface acoustic waves. So this is your reference value. Okay. Now you have an unknown analyte. When your bioelement is interacting with the unknown analyte, so-called analyte, what we do is that we have a reference value already. We get output value. So we see how much of deviation is we are getting from reference value to the output value. And based on that, we say that whether the signal is getting increased or the signal is getting decreased. Okay. So this way we can recognize depending upon the changes that are happening at the mass level. Okay. So this is one type of sensation, and we can use the sensations even for some of the technical devices, like people who have hearing aids and all. They usually will be going ahead with the devices which can enhance the sound, and these devices will be usually made up of this transducer called as resonant biosensor. So this is first category. Coming to the second category, optical detection. These are the most widely detection systems in day life. A common man usually comes across सबसे ज़्यादा detection common man के लिए यहाँ से होता है, optical detection पे होता है. Why? Because most of the chemical compounds in our biosystem are having different phenomena with respect to light. Okay, so we will be using usually 2 phenomena. One is absorption phenomenon, and the other one is called emission phenomenon, or the second one is deflection phenomenon. So we depend upon this. And we will be able to do the recordings based on that. Okay, so majority of the times we go for absorption based. So have a chemical, let's say you have photosynthesis में so and so so-called phytochrome photosystem is able to receive the photons from red region. So what we do, we give a wavelength of around like 700 nm. okay, or 720 nm, and then we will be able to record the changes that are happening at the red wavelength. Okay, so these sort of devices usually are called as spectrophotometric devices. Okay, and in most of them, what do we use? We use photodiodes. Okay, so we use photodetector. So how is a photodetector can establish in simple systems? We can establish using a silicon wafer. This silicon wafer can actually diffract the light. Okay, so we completely rely on this phenomenon called as optical diffraction. So in this optical diffraction method, we will be able to set a reference value at this point. यहाँ पे हम detection करेंगे. Okay, so you have your biological element bound here. So whenever this biological element is binding to biological compound is binding to your analyte of unknown sample, you will be getting a change in the pattern here. ये जो optical detection में आपका जो wavelength है वहाँ पे आपका reference value से change आपको मिलेगा, and that's what you will be recognizing, and that's what you will be getting it. Okay, so this is like one of the most common methods that we adopt. So if you're going to come across the devices where we actually employ optical detection systems, regular day-to-day uses में हम use करते हैं. Nowadays, most of the smartphones that we are getting basically has a touch screen. Okay, so you have a fingerprint sensor available on your touch screen only. That relies on the principle of optical detection systems. Okay, your biometric. आप लोग जब kit में आएंगे, I don't know about other places, but when you get into the kit, in kit we give the attendance using biometric systems. Okay, so they take the fingerprint reading. So fingerprint, every one of us have a different pattern. So depending upon the optical densities that are recorded, a close value is being recorded, and when we place our fingers on the biometric devices, if the value matches, then it records our attendance. Okay, it's not just for recording attendance. We even use it for the so-called opening and closing of certain lockers or gates. Okay, same way you can even look at the biometric thing available on your laptops. Okay, fingerprint sensors available on your laptop, or retina scanners available on your laptops or any other areas. So all of these basically rely on the so-called optical detection systems. Okay, even the smartwatches that we wear basically has different types of optical sensors that will be giving you different outputs. Some of the outputs is related to the number of steps that you have taken in a day, and some will be giving you about as of now in the current scenario, many of the watches are coming out with the inbuilt SpO2 sensor. Okay, so this way, and in during Corona, most of the so-called people are purchasing pulse oximeters. So that also relies on the so-called optical detection systems. Okay, so all of these compounds that what a common man is using nowadays, many of them do rely on optical detection systems. Okay, so this is about optical biosensors. Now coming to the thermal detection biosensors. So some of the reactions do take place where they see a change in the temperature, heat. Okay, so in some cases you can see an exothermic reaction. In some cases you see an endothermic reaction. So when you know that particular reaction can be exothermic or endothermic, in that case you can use a transducer, especially thermistors, which can convert thermal form of energy into electrical form of energy. Okay, so the binding of an analyte, when an analyte binds to this so-called biological element, that brings about a change. That change can be endothermic in nature or exothermic in nature, which means in some cases heat loss happens, in some cases heat gain happens. Okay, so this heat loss or heat gain can be recorded, and that's where we are able to get the data. Okay, one of the most simple device that we have been using for a long time is thermometer. The thermometer will be able to recognize the surface temperature. Okay, so digital thermometers that we are using now is one of the classical examples that I can give you. And other than that, there are many other devices which we usually use it for recording pesticide availability or any parasite activity in the body. Okay, so this is about thermal biosensors. Now coming to the ion sensitive biosensors. So what do they do? So some of the reactions are completely dependent upon the changes in the ionic concentrations. So in such cases, what we can do is that we can actually take a biological sensor and record the changes in a specific area, and you call this area as field. Okay, so such kind of detection is called as field effect transition. Okay, so the devices that we use for detection of ions are field effect transistors that are ion sensitive in nature. So we have two different types of devices again in this. One is ion sensitive field effect transistor, and the other one is enzyme field effect transistor. So both of them will be able to, ion sensitive kya hota hai, that will be selectively choosing only certain ions. Like let's say if you want to go for calcium ion measurement, or let's say you want to go for magnesium ion measurement, or a sodium ion measurement, or potassium ion measurement, then you should be able to use only that selective FET. And this FET will be able to record only those ions. Okay, this is one thing. And then you have another kind of a transistor called as an ENFET, enzyme field effect transistor, which primarily relies on the generation of hydrogen ions. So we use these for detection of pH, basically, okay, in the living system. So this way we have two different types of ion sensitive field effect transistor based biosensors. Okay, they will be able to detect the ionic concentration in a certain media. Then comes electrochemical. So electrochemical. So you have certain reactions where the ions or the electrons tends to get produced or consumed, and this bring about a change in the electrical properties of the solution. So what we do here, we detect the changes in the electrical properties of the solution and we sense out. So depending upon what we are measuring, like if you are measuring the current, then it's amperometric. If you're measuring the potential of the media, then it's potentiometric. So we have different types of biosensors available on that, out of which the highest utilized will be amperometric. So these kind of devices, we use them in majority of them will be used in the medical systems. मतलब hospitals में जब testing चल रहा होगा, so वहाँ पे optical systems का इस्तेमाल ज़्यादा ही किया जाएगा, उसके साथ electrochemical का भी इस्तेमाल बहुत किया जाएगा. So these two are the widely used biological sensors in the medical field. One is optical, the other one is electrochemical. So in electrochemical, we have one example of amperometric detection. That's glucose biosensors. So as of now, today we are able to see many companies coming up with sugar checking devices, okay, which are portable, handy. So what do they have? They have these probes. You can actually purchase the probes. The probe will be having an enzyme called as glucose oxidase, as you can see here. So those glucose oxidase will be able to act on a glucose, and it converts glucose to gluconic acid. Okay, so this enzyme undergoes reduction when this conversion happens, and it will be emitting you so-called oxygen, and it will be giving you electrons and hydrogen as well, protons as well. Okay, so what we can do is that at that point of time, as soon as immediately you get it, you can actually look out for the changes in the electron density. I mean, यहाँ पे जो electrochemical property है वो आप record कर सकते हैं. So you can use an electrode. Usually platinum electrode or a graphite electrode can be used. So when you're using a platinum electrode, the advantage is that it can even recognize the changes happening because of oxygen levels. So if you can see the oxygen levels in a higher content, that means that the glucose content is too high, and so your oxygen liberation is also too high. Okay, so this is one method. Okay, and the second method is you can also look at hydrogen peroxide levels. So hydrogen peroxide usually will be very less when you have high glucose, and when you have a low glucose, okay, so this conversion doesn't happen, and so you are left with more of hydrogen peroxide. Okay, so this way you can either go for recording of oxygen, or you can either go for recording of hydrogen peroxide. So you see the red one is indirectly proportional, and the blue one is directly proportional product. So glucose levels can be matched either with your oxygen indicator or either with your hydrogen peroxide indicator. This shows direct proportionality. This shows indirect proportionality. Okay, and eventually the glucose oxidase further will be oxidized back, where it will be utilizing the intrinsic available oxygen, and it will be getting back to its normal shape. Okay, so immediately when you are putting a probe and when you are giving it to the sensor will be able to record the changes that are happening in the probe area, the solute, or they also can be able to record the oxygen level, or they will be able to record the hydrogen peroxide level. So depending upon what kind of sensor you're using, you can use platinum usually electrode to record the oxygen levels, or you can even use a graphite electrode as well. Okay, so इस तरह हम किसी आदमी के blood में कितना शुगर लेवल है वो पता करने के लिए we can use this sensor called as glucose biosensor. So this is an amperometric device. Okay, so here we are actually looking the current in terms of electron flow. Okay, now coming to the uses, where can we apply? पहले हम हर केस में applications देखते आ रहे थे, but let's look at other applications. Okay, let's see the DNA detection methods. Okay, I'll give you just one example. I'll just give you one example. Now the entire world is suffering from pandemic, so we have COVID. So in COVID, we are going ahead with two reactions. One is rapid test, and the other one is RT-PCR. Okay, so here we have a principle that relies on the RT-PCR. This is called as polymerase chain reaction (PCR). So what we do here is that we know an analyte, okay, which is basically a DNA. So this analyte basically has many genes in it. So what we can do is that we can take the organism-specific gene, and we want to see whether when we give a probe to it, whether the probes are able to multiply it or not. अगर multiply कर पा रहा है इसका मतलब वो gene वहाँ पे है और वो active है. Okay, so that's what it determines, and based on that only we will be able to say whether the person got COVID or not. Okay, so let's say there is some gene X in the coronavirus. Okay, so a person, let's say you have got a sample from an unknown person, and you want to see whether the person basically have a COVID or not through using PCR. Okay, so we have an advanced method of PCR called as RT-PCR, which is real-time polymerase chain reaction. So in real time, what we do is that we are going to use a fluorescent dye called cybergreen or a Tachman probe. So the cybergreen, what it does is that whenever your DNA gene gets detected, and whenever उसको copy जो DNA polymerase का जो activity चल रहा होगा, so you have a synthesis of double chained DNA. So this fluorescent dye goes and bind to the double chain DNA, and it starts to give you a fluorescence, which is basically green color. So what we do is that we do a detection at green wavelength, and we get the value. If you get hits as early as the 5th cycle or 7th cycle or 10th cycle, you see that okay, the so and so sample that I got is getting a hit as early as possible, which means that the gene for the COVID is present. Okay, and let's say somebody is not even getting a hit after 25 cycles. We say that okay, even after 25 CT value of 25, we are not getting a hit, which means the cybergreen is not getting activated, which means there is less chances of availability of the gene in the sample, and so the report will be negative, COVID negative. Okay, so this way we have the detection of DNA using this so-called process. Same way we have other processes also. We have force amplified biological sensors, we have force differentiation assays, we have bead array counters. All of them again are technically like long processes. We just remember the names of these processes. Okay. So what do we use them for? We use them basically for detection of DNA, especially in case of detection of harmful agents like anthrax, botulinum, food poisoning agents, or any other infectious agents that are available in an analyte. Okay, so जब भी हम हमारे analyte में कोई देखना चाहता है कि do you have any harmful stimulating agent in it? In that cases, these assays are good. So this is about DNA detection. Coming to detection of pollutions. Okay, so you can use different microorganisms to identify the environmental pollution. Why? Because certain microbes really grow faster when the conditions are polluted. What we can do is that we can use these pollutants, and these pollutants can be done with testing. We can do two different tests. One is reporter assay, and the other one is immunoassay. Let's begin with immunoassay. Immunoassay is a colorimetric based detection, where you have a so-called microbe with its biological compound, which can interact with the pollutant. So what we can do is that let's take an unknown sample in which you want to know the level of pollutants in it. Okay. So what we can do is that we can take this biosensor fitted onto this bioelement. So this bioelement, when it interacts with the analyte, it will be changing its color. Okay. उसका जो physical properties है वो change होता है. So we can use color emitters, and then we can see report that okay, see ज्यादा कलर आ रहा है, इसका मतलब ये particular sample में pollution काफी ज्यादा है. अगर कम कलर आ रहा है, then we can say that okay, this particular solution or this particular sample has less pollutants in it. We can go ahead with whatever the process we want to do. Okay, so that's one kind of detection. The other detection is through reporter assay. So what do we do in reporter assay is that we have a cell, bacterial cell, which we are going to engineer. Okay, मतलब genetically हम इसको modify करेंगे. So what modification are we going to do? हमारा एक gene है जो pollutant को express होने से activate होता है. So this gene, to this gene, we are going to now add a reporter. So what is this reporter? Basically, it's a Lux gene. So what does this gene do? Whenever an activation happens, the products that are going to come out will get oxidized and they start emitting light. The process is called as bioluminescence. Okay, so we use fluorimetrics or we use optic devices to record the intensity of the light that is coming out when this reaction is happening. Okay, and based on that we can say अगर सबसे ज़्यादा glow आ रहा है इसका मतलब pollutants काफी ज़्यादा है. अगर glow कम हो रहा है या light intensity कम है इसका मतलब pollutants काफी कम है. Okay, so this is a kind of a detection. So here you can observe that we are actually going ahead to recognize the non-biological phenomenon as well, मतलब non-biological partners as well. Okay, so here we are targeting pollutants. So this is one kind of analysis that we can do. Then food. Even in food also we can use the devices for detection of lot of material. Like let's say on the day of New Year or any other day of festivals, police do come out onto the road with a lot of these devices. Okay, so these devices will be able to identify the alcoholic content that a person has consumed. Okay, usually a human body will be able to generate some alcohol because of the so-called the excess muscular activity. Okay, and on top of we have something called as anaerobic respiration. We do that. Muscles do that. Okay, and on top of that we have several other bacteria available in our body which can do fermentation. So we tend to have a limited amount of alcohol already generated in our body. Okay, so when we consume alcohol, बाहर से जब हम alcohol consume करते हैं, that will be able to emit certain vapors, and these vapors are our analyte. So what we do is that we have a reference value which says that okay, up to this level you can leave the person. If the level crosses, you should hold on to the person. The person should not take part in driving. Okay, so the police, what do? They come up with these devices where you have to blow into the pipe. So when the vapors hit the reference sensor, and if it crosses the reference value and if it gives a beep, वहाँ पे भी हम देखते हैं reading जो आता है, that's coming out as a digital input, right? So that's a conversion. So here we are converting the gaseous form. We are detecting the changes that are happening at the gaseous form, and we are going to get the reading in analytical format. Okay, so so here we are able to see whether the person has really drunk more alcohol or less alcohol, and based on that we either allow him to go ahead or we either allow her or him to stay back. Okay, so this is like one example I can give you, and other examples like in case of foods, as of now many of the companies are coming with a lot of nutritional information data. It's mandatory. जब भी आप कोई food product दे रहा है बाहर की तरफ, you have to give, especially the packed food, you have to give the content. कितना content में कितना protein है, कितना fat है, whatever. Like you take a milk, you take a biscuit packet, you take Lay's packet, everywhere you have this nutritional information, right? So when you think that somebody is actually playing with the so-called data, they are giving you a manipulated data. Okay, like I will give you an example. Let's say we have some 60 plus students in the class as of now. Okay, where is this? Yeah, let's say we have some 60 plus students in the class. Let's say just an example. Let's say Shagnik Roy is one person who wants to build his body well. Okay, so let's say he wants to look so muscular. So he has to do a lot of exercise. At the same time, this fellow has to come up with good amount of protein-based diet. Okay, let's say. Then let's say we have this Ankit, who has actually come up with a noodles company. And let's say usually noodles में हमारा protein content काफी कम होता है. Okay, but let's say Shagnik Rai likes noodles very well because of his bodybuilding activity. He has to quit the noodles, eating noodles. Okay, let's say Ankit now comes out with a product which says that, whose nutritional information says that for every 100 grams of noodles I'm giving you, I have 50 grams of protein in it. Okay, So which is basically a very luring offer for most of the noodle, lovable fellas. Okay. So let's say Sagnik feels very happy and thinks that okay, इसमें protein है तो हम noodles भी खा सकते हैं. At the same time, I'm getting protein as well. Okay. Let's say this fellow is doing all sorts of activity and he's taking only noodles for a quite period of time, and let's say he's not getting the expected result. Okay, so he can feel something fishy about the product. So what he can do is that he can take the product, give it to the so-called food-based laboratories, and the laboratories will be able to detect the carbohydrate level, the protein level, fat level. They have different methods. They have flow injection system, they have HPLC. So depending upon these methods, they will be able to give you the data. So what you can do is that you can correlate कि ये जो अंकित का जो फैक्ट्री जो डाटा दिया है वो सही है या नहीं है. Okay, so if you get a false data saying that okay, 100 grams of Maggi basically have only 5 grams of protein, and this fellow is claiming 50 grams, straight away you can go sue him. Okay, you can put file a case on him and you can actually sue him. Okay, so recently, like in last 5 to 6 years, you might have seen most of the food-based companies are actually onto the court, onto the media. Okay, this is all because of these kind of things. Okay, so we can use it for quality analysis, we can use it for quantity analysis. Okay, जैसे food में कुछ लोग adulterants use करते हैं. They instead of using what is it, mirchi powder, they can use red color, simply red color. Okay, that we use it for playing Holi. Same way, haldi में भी color component use कर सकते हैं. And condiments and spices may also there are a lot of adulterants that are being used. Okay, so for detecting of these many number of false things in the food, you can actually go ahead with the biosensor industries. Okay, so the biosensors are playing a very crucial role in the food industry, and there are many laboratories established to detect the levels of the contents in the food. Okay, so this way we can use the biosensors in many areas. Okay, and they are very applicable. So as of now, people are actually coming up with a good amount of biosensors. Okay, we have millions and millions of biochemical reactions, out of which maybe hundreds might have got a hit. Okay, and there are more biosensors to be evolved in future. Okay, as we go ahead with the growing conditions, growing necessity, there is never ending dearth of necessity. So this is like one of the hot topics where the people can work out, and they can integrate the biological technology with the so-called engineering technology, and they can come up with such kind of marvels. Okay, so this is all about biosensors. In case if anyone has any doubts, let me know. In case if anyone has any doubts, let me know. So I'll be sharing the slides once we are done with the next topic. Also, I'll share all of these slides with you all. Okay. So principle, आपको diagrams कौन सा practice करना है? See, you can actually practice this. This is very much important. Elements of biosensor is very important. When you are asked about components of biosensors, then you can actually draw this one. This diagram is very important. So you have all the components. You have analyte component, you have a bioelement, you have a transducer component, and then you have detection component. Then coming to the types, we have different types. You can refer to the book. In book, much explanation has been given so that you can understand better. Okay. So yeah, this diagram is very much important. The glucose biosensor diagram is very much important. Technique is very much important. You can look at the diagram from here, or you can look at the diagram from the textbook also. You can practice this. Then DNA detection, all of these. Yeah, this diagram is also important. Biosensor in pollution industry. This is not available on your textbook. This is available on slide only. Kindly mark this as important, and you have to practice it from slide only. Then that's it. Okay. So this is all about biosensors. In case if anyone has any doubts, let me know. Okay, fine. If there are no doubts, we will hang up for a while and we will catch up again at 11:05. Okay. We'll catch up again at 11:05. We'll complete the next topic and then we can discuss. Thank you all.
Thank you,
Thank
sir.
you, sir.
Yes, sir. Thank you, sir.
That's about molecular motors and machines. So here, this presentation will be having a PPT followed by I'll be showing certain YouTube links and I'll be showing certain videos for better understanding purpose. Okay, so yeah, let's begin the topic. So in the beginning, we have actually learned that why actually we are learning biology. What is the need of learning bio and engineering and all. So I have already told you that the biological systems are like the marvelous what do you say kind of models to study at the engineering angles. Okay, so if you're going to look at the cell itself is automated, and the way it performs will make it look like a miniature factory made up of different types of machines and motors available in it. Okay, for example, I'll tell you. Let's say we have this, we have come across this process called as RNA splicing or RNA editing. RNA editing में हम देखते हैं कि बहुत सारे protein molecules आके DNA junctions जहाँ पे है वहाँ पे cut करके we are eliminating the introns and we are joining up the exon. Right. So when we are doing that, so these kind of machines basically are so-called RNA-based machines. Same way you have translation, you have replication. All of these basically depend upon your nucleic acids, DNA or RNA. Okay, and how do they work? They work in congregation of different protein molecules, and these protein molecules will be bringing about a work. It can be a synthesis work, or it can be a cut and paste work, or it can be anything. Okay, so same way you have many such kind of machines available for production purpose, for repair purpose, for cleaning a purpose. Okay, so if we're going to look at the cell, we have diverse range of activities happening in a cell. Okay, so like we have generation of voltage gradients across the membranes using the so-called ion channels. You have nano assemblies which will be making the working of your so-called majority of the nano assemblies usually we see forming at your DNA or RNA, as you can see here. And you have many other microscopic assemblies that will be making the cell to do certain motorized activity or mechanical activity. Okay, so out of all of these molecular machines, we are going to study about two different categories. One category are rotary motors, and second category are stepping motors or linear motors. Okay, so beginning with rotary motors. So what do they do basically? So these are assemblies of different protein molecules, which tends to exhibit a rotary motion depending upon the chemical gradients. Okay, so depending upon the chemical, so what gradient are we depending upon? You may have a positively charged ion, or you may have a negatively charged ion. So based on the availability of these ions, there are certain conformational changes that happens at the molecular level, and this bring about a change in the mechanical activity. Okay, so these kind of motors are called as rotary motors. So they do a rotational activity. And on the other hand, you have stepping motors or sliding motors. So what do they do basically? They have a motor domain. So they utilize the energy, and they will be just walking along, or they will be making the so-called components pull towards them. Okay, so this way you have two different motors, rotary, stepping. So rotary motors, let's begin. We have two different models of rotary motors available in the course. One is F0 F1 ATP synthase, and the other one is Bacterial Flagellum. Okay, so let's begin with the F0 F1 ATP synthase. So this is a small motor which basically has two components, an F1 component and F0 component. F0 component is the component that is available on the mitochondrial membrane. So mitochondria has two membranes, outer membrane followed by inner membrane. The inner membrane will be having this component called as F0, as you can see here, this one. And the outer, beyond the inner membrane, you have the so-called matrix or the medium of the mitochondria. So in the medium of the mitochondria, you have a static component, which also is a rotor, but it doesn't rotate as much as this rotor moves. So this component is called as F1. So if you're going to look at F0, what is F0 made up of? It's made up of A element, where 4 to 5 proteins of A will be combining and making an inlet and outlet for hydrogen ions. You can see this is an outlet and this is an inlet. Okay, and this configuration is in match with the rotor. Okay, so a rotor, this rotor is basically made up of C proteins. These C proteins are the proteins that exhibit either an oxidation or reduction with respect to the protons. Okay, matlab either they accept the proton or either they remove the proton. So depending upon the acceptance and rejections, the site where this changes happens will be in contact with this A ring. So this forms your so-called F0 fragment. Now connecting to our F0, we have F1, which is made up of 3 units of alpha. You can see we have 3 alpha and 3 beta, 1 gamma, 1 delta, 1 epsilon. So gamma and epsilon will be forming the stock. So whatever the rotational force is being carried out here is being generated this way. Okay, so a torque is generated in a downward fashion. Okay, and that will be like giving us the force for a chemical reaction to happen. Okay, and on top of that, you have the peripheral supporting partners coming from delta and coming from B. So you have A, B, C, alpha, beta, gamma, delta, epsilon. So these all are the components of your F0 F1 rotor. This diagram is very much important. You have to practice it. So how does the working happens? How does the working happens? For that, I'll show you a small animation.
Sir, can you show the diagram once more?
I'll show it once more. Let me just show you the animation. So once we are done with the animation, then I'll get back to the so-called presentation part and I'll be displaying that. Okay. एक बार मैं animation आपको बता दूंगा, उसके बाद आप देख लीजिए. Is this visible, everyone? Is this animation visible?
Yes, sir.
Yes, sir.
Okay, this is without sound. I'll be just briefing what's happening there. So you see, this is a mitochondria. Okay, so not just mitochondria, even on your bacteria or even on your chloroplast also, we have these kind of arrangements that takes parts in movement of hydrogen ions or sodium ions. Okay, so in case of eukaryotes, we depend upon hydrogen ions only because we have electron transport chains and we have light reactions which completely are dependent upon the generation of protons. So what happens here is that once an electron transport chain series goes on, you have accumulation of protons in this space. So you can see this is your outer membrane, this is your inner membrane. In between, you have a space called as intermembrane space. Okay. So in the intermembrane space of mitochondria or a chloroplast, you have more accumulation of protons, H plus ions only, and that leads to generation of a force called as proton motive force. And this proton motive force activates the inlets of your A complex, which makes the proton to enter into the A complex and bind to the empty C protein. So this is a reduction reaction. यहाँ पे आप देख सकते हैं, यहाँ पे C protein में ये moiety जो था वो empty था. So your so-called proton has used this inlet and it has bound here. As soon as this happens, the backward C protein undergoes a conformational change, emitting one proton outward. You see, yeah, that one. Once again, see, you have a reduction simultaneously followed by an oxidation, and a rotation happens. So this way, okay, this way the rotor rotates, and as the rotor rotates in the matrix space, you have the epsilon and the gamma units acting, and these mechanical force is being utilized. मतलब वहां पे भी ट्रांजैक्शन हो रहा है जहां पे आपका मैकेनिकल एनर्जी को हम केमिकल एनर्जी की कन्वर्शन कर रहा है. So where is it happening? That's happening at this F0 F1 complex. So what's happening here in the F1 complex? You have 3 alternate sites. Okay, so at every alternate site, for every 4 protons that are actually coming in, you have a adenosine diphosphate picked up from the area called as matrix. So ये matrix का ये जो ये free ADP है और ये inorganic phosphate जो है, that is being taken in and that's allowed to couple up. So the force couples up the adenosine diphosphate to a phosphate, making it to form adenosine triphosphate. So this force goes on until the concentration gradient here disappears. Okay, मतलब यहाँ पे जो proton motive force आपका जो generate हुआ है, वो जब तक कम हो जाएगा तब तक ये process होता जाएगा. So you see, now here we have ATP being synthesized, and next backward, वहाँ पे synthesis हो क्या आएगा, फिर यहाँ पे synthesis होगा. So this way you have generation of ATPs every time. Okay, so ATP generation depends upon how many number of protons are acting and how many number of C's are there. So C usually in case of mitochondria is 12 in number. So which means that for making 3 ATP molecules, you require so-called all the 12 proteins to rotate. Matlab for every rotation, for every small moment, you are going to have an angular momentum of 30 degrees. Okay. Why? This is 360 degrees. This is made up of 12 C proteins. So for every oxidation and reduction, you are getting an angular momentum of a small momentum. मतलब एक protein का moment हो रहा है, which means that you have 360 by 12. So that will be giving you 30. Okay. So this is in case of mitochondria. In case of chloroplast, the proton binding efficiency vary. The number of C proteins vary. In case of bacteria, also we have a similar kind of a complex available, which will be performing its activity on the plasma membrane. Okay. So, and in case of bacteria, additionally, it depends upon the sodium ions also. We have 2 types of ions, sodium ions, protons. In case of eukaryotes, it's only protons that are used. In case of prokaryotes, we use both sodium ions and we use both hydrogen ions as well. So this way, we are going to get the energy being generated from the mitochondria, and from the mitochondria, ये जितना भी energy आ रहा है, out of all the organelles in the entire cell, this way of generation is the fastest way, and this way of generation is the highest output as well. And that's the reason why we are calling this entire mitochondria as the powerhouse of the cell. Okay, I'll share this animation with you all. I'll share that animation with you all. So that's about the functioning of a rotary element in mitochondria. So there we have seen that it is generating the energy molecules, which are nothing but your ATPs. Okay, so that's the smallest rotor. So this is the diagram. You can practice this diagram, or you can have practice the diagram from the textbook also. मतलब यहाँ पे मैं 2 diagrams को दिया हूँ. One is this one, and the other one is this one. So this is a 2D diagram, and this is a 3D diagram. You don't have to practice this one. You actually can go ahead with this. This is easy, and this has all the components being mentioned properly. Okay, या तो आप इसको practice कर सकते हैं, या तो आप textbook वाले diagram को practice कर सकते हैं. So this is about a small rotary element, and this can be reversed. Okay, इसका जो activity है वो reverse कर सकता है depending upon the availability of ATP here, depending upon the proton gradient in the so-called matrix, it can undergo reversion. Okay, usually the frequency of the rotation will be in a forward direction only. मतलब यहाँ से यहाँ तक protons का movement हमेशा होता रहता है, क्योंकि हमारा जो ATP generate हो रहा है वो utilize होते जाता है cell में, and that's the reason why this is always a forward reaction. But there are events where it can undergo backward firing as well, and there this is now being utilized as the inlet, and this will be used as an outlet. Okay, so this is one kind of an activity, rotary based activity that we see in the nature. Coming to the second component, the bacterial flagellum. This is a congregation of several rotors. Why? Because bacteria में देख लीजिए, you don't have simply plasma membrane. You have plasma membrane, you have a peptidoglycan gradient, you have a lipopolysaccharide layer, then you have outer so-called cell wall. तो इस तरह bacteria में बहुत सारे layers हैं. तो हर एक layer में you may come across a rotor, as you can see here, or as you can see here. You can use this diagram, or you can use this diagram. Both of them you can use. Okay. So मैंने यहाँ पे 3 diagram mention किया ना. Yeah, 1, 2, 3. This is 2 and this is 3. So you can go ahead with this diagram, or you can go ahead with this diagram. दोनों drawable हैं. These two are easier to draw. So if you're going to look at the bacterial flagellum, so you look at this. This is the cross section. Okay. So when you enlarge it, it's going to have 3 components. One is the basal body, which is nothing but the congregation of all the rotors. Followed by that, you have a hook, which is the connecting link between the filament and the base body. And then we have the filament, which is basically the functional component of your flagellum. Okay, so how does the energy generation happen here, and how does this will be making use of it? That we are going to study now. Okay, so if you're going to look at the lowermost rotors, the lowermost rotors are accompanied by mode A and mode B proteins. That green color proteins that you're seeing here, they're called as mode proteins. They are the one which undergo conformational transitions depending upon the gradient here. यहाँ पे जो बैक्टीरिया में सोडियम आयंस की वजह से या पोटेशियम हाइड्रोजन आयंस की वजह से क्या होता है, these two rotors can rotate, and their rotation is enabled by the movement of these two, mode A and mode B proteins. So we have mode proteins, so that will be generating the motorized activity of the rotors. Fine. Now the motor activity does not always go in a single direction. It can go in two directions. One is a clockwise direction, as you can see here, and the other one can be a counterclockwise direction. Okay. So clockwise direction and counterclockwise direction, ये swapping कैसे होता है? That happens because of the core proteins that are available here, which we call it as FLI proteins, FLI proteins. What they do, they make the orientation vary. Sometimes they allow the flagellum to undergo clockwise orientation, and sometimes they allow the flagellum to undergo counterclockwise orientation. Okay. So in conjunction with these two things, you have a rod running in between all the rotors. This rod is very crucial for a synchronized activity. मतलब ये नीचे की तरफ जो भी rotations हो रहा है, ऊपर का rings में भी वही rotations दिखाई देगा. And how are we able to bring out the synchrony? Because of this rod. So the rod connects all the rotors from lowermost position to the highermost position of the basal body. Okay. Once the interconnection is established, then the same rotation is being carried ahead into the hook. And at the top of the hook, you have self-assembling complexes called as flagellins. These flagellins will be making up this long tail-like structure called as flagellum. Now, the base that is coming into the hook also enters into this so-called filament. Okay, making the filament actually to undergo rotations. And this way, when the voltage on the membranes crosses 150 millivolts generated because of hydrogen ion or Na+, okay, you have the rotation being happening. And we have two rotations, as I said earlier. The rotations will be either generating a torque of like 8 to 16 torque, 8 to 16. Okay, and at the rate of 16 torque, they can travel as fast as 1000 hertz. I mean, the filament can actually rotate at a speed of 1000 hertz. Okay, which makes the bacteria to travel at the rate of 70 times of their body length. Okay, and in this way, the so-called the speed attained by the bacteria with respect to its body weight, body length, is very, very high compared to any other gauges in the nature. These are the fastest. Okay, so where do we get 8 torque then? This usually 16 torque, we usually get it in clockwise direction, and the torque of 8, we usually get it in anti-clockwise direction. So clockwise and anti-clockwise होने के लिए, the FLI proteins that are available here are very much important. You can see in between you have actually got these proteins called as FLI proteins. कहाँ पे गया? Yeah, this one, the blue, these ones. Okay, so they make, depending upon the senses that are coming from the flagellum. Flagellum actually does two jobs. One is to sense danger, and the other one is to sense the nutrients. Okay, so whatever the sensation the flagellants are giving downward, the FLI proteins will be recognizing that, and they will make the rotor to either undergo clockwise rotation or to undergo counterclockwise direction. Okay, so clockwise कब होता है? When the bacteria wants to move from one area to other area, then the rotation is clockwise. When the bacteria wants to settle down at a particular area and eat the food or take the nutrients inside, then the rotation is counterclockwise. So counterclockwise, remember, it's basically when the bacteria is settling down. It's basically when the bacteria is munching on its food. And clockwise, when the bacteria is running away or when the bacteria is moving from one location to other location, it's clockwise. Okay, so this way you have the bacterial flagellum been functioning. And coming to the arrangement, some of the bacteria may not have a flagellum, some of the bacteria may have flagellum, some of the bacteria may have more than one flagellum, but the positions can vary. So depending upon the flagellar availability and depending upon their positions, you have 6 categories of bacteria. The first category are Atrichus, which means that there is no flagella at all. Second category is Monotrichus, which means that there is only one flagellum on the entire body of bacteria. Third category is Amphitrichus, which means that there are 2 flagella. Both the flagella are available at both the ends, 2 poles basically. This is Amphi. Then Lophotrichus, which means we have more than 2 flagella, but all of them are available on a single pole of the bacteria. So this kind of arrangement is called as Lophotrichus. Then we have amphilophore, which means that you have many flagella, but their arranging points are at two different points only on the entire body of bacteria. 2 points are the areas from where they generate, so making them look like two tufts. So this way you have two tufts of bacteria, two tufts of flagella available on two sides. That's called as amphilophore. This is mono, this is lopho. This is amphi, this is amphilophore. This is atricus, and then comes the last one called as peritricus. Peri means you have many flagella, and all of them are available on the entire body of the bacteria. That's called as peritricus. Okay. So now, for better understanding, let me just share a small.
Yes, sir.
Yeah. Tell me. Yes, tell me.
No, sir. I was just.
So look at the tab. Yeah. So this is how bacterial flagellum is working. You can see it's actually rotating. Okay. Whatever the rotations are being generated here are carried forward. Why? Because we have a central rod. The rod is making the same torque move across this hook, and from here itself the rotations are carried forward onto the end. Okay. So यहाँ पे आप देख सकते हैं, it's everything is rotational. Okay. Now you can see this is going in an anti-clockwise direction. So यहाँ पे आप rotation देख सकते हैं, वो काफी slow चल रहा है. Now when it turns onto the clockwise, the rotations are going to be pretty fast. Okay, so this turning of counterclockwise to clockwise or clockwise to counterclockwise is assisted by your FLI proteins, FLI proteins. Okay, so these are your mode proteins. They are very much important for the rotation of these lowermost rotors. इसका जो speed है, वो speed regulation कहाँ से होता है? इन लोगों की वजह से होता है, from these components called as mode A and mode B proteins. Okay, and उसका जो orientation है, that is dependent upon the FLI proteins which are located here. So this way you can see you have a rotor here, then you have a rotor here, then you have a rotor here. This is like an entire congregation of gears and stators and all, right? So this way your bacterial flagellum will be working. So this is about the so-called rotary elements. Now coming to your linear motors. So let me just share the PPT. Why it is not getting shared? Yeah. Kindly look at this. So is my presentation being shared?
Yes, sir.
Okay, good. So let's look at the linear motors. Now looking at the linear motors. So linear motors, they unlike like the rotary motors. Rotary motors में हम क्या कर रहे हैं? हम power को generate कर रहे हैं, depending upon the chemical gradient. We are generating the power, and we are either storing the power or we are either utilizing it for the mechanical activity. But here, what we do is that we don't have any chances of generating the power. Whatever the power is available on your cytoskeletal elements or on your so-called cytoplasm, that has been consumed up, and they bring about a change in the mechanical activity. So these kind of motors are called as linear motors. So we have the common configuration for all the linear motors. There is a motor domain that you can see here in all the three cases, followed by there is a central stalk which is made up of amino acids. It can be a short peptide or it can be a long polypeptide. As you can see here, this is a short peptide. So usually we have a stalk. The stalk at the end is flexible. Okay, in every case you can see this is flexible, which means that it can make certain moments. Okay, and at the end you may or may not have a light chain. This chain is basically for interaction with the cargos. Okay, cargo, when you want to do transportation, at that point of time you require cargos. Okay, So we have three different types of activity that can happen. One is cross-linking, or one is sliding activity. So you can see this sliding activity in case of muscles. Okay, and the other one is this porter-based activity. This is the coolie that you usually find on your railway stations or your bus stations, right? So they will be transporting the cargo from one particular area to other area. All right. So we have these two types of activities. So how do they do this activity? They rely on our cytoskeleton. We have a cytoskeleton, right? The cells basically have a cytoskeleton. So the cytoskeleton is made up of three different types of segments. One is microtubule, which are stouter in nature, as you can see here. These are your tubulin proteins making a tubule. Then you have a microfilament, which is made up of a protein called as actin. So these two tubules are the filaments, play a very, very big role in the movement or in the motor function. Okay, so let's begin with one of the molecular motor called as myosin. So myosin is a motor that is available in your muscles. Okay, so when you are actually coming across a cytoskeletal element, it has two ends, a negative end and a positive end. So negative end, matlab, this is the end from where the production basically has begun, and positive end, matlab, this is the growing end. Okay, so a starting end is always termed as minus end, a growing end is always termed as positive end, be it a microtubule or be it a microfilament. Okay, so what happens is that the myosin head, this is your motor domain. The motor domain will be able to break the ATP. When the ATP is broken, energy is liberated. Using the energy, this will be binding onto your microtubule. So when a microtubule is bound, that is being dragged. मतलब यहाँ पे हम क्या कर रहे हैं, हम उसका पुलिंग कर रहे हैं, जिसके वजह से क्या होता है, आपका साइटोस्केलेटल को एलिमेंट्स को अगर हम पुल करेंगे, what happens, that leads to contraction. मतलब हम हमारे muscles को देख सकते हैं, they can contract and relax, which means that the volume of the muscle can actually go down and get back to normal. So how does the volume of the muscle get shrunken, or how does it go down? Basically, it's happening because of this motorized activity. So here we have this motor domain that is dragging your microfilament. Okay, and to understand it better, let me just share a small video again. So just have a look at this. So these are your actin filaments. These are your actin filaments. These are your muscular fibers. So these fibers basically have the protein called as myosin. The myosin will be making these actin cytoskeleton filaments to undergo movement. So they pull them. You see? So when they're pulling, what's happening? Your entire muscular volume is actually getting low. Right? And once the activity is done, they will be releasing it from the pockets of actin, and the actins will move backward, leading to relaxation. So you have a contraction followed by relaxation happening. So you can see here, once your ATP is split, and once the calcium binds here, then the pockets are free here. Now the myosin can go and fit into these pockets of actin. So the myosin binding sites are active now. It binds to the actin pockets. The heads are inserted into the pocket, and this will be dragged backward. So when it is dragged backward, you can see the ATP will be leaving. So the power generation, whatever has happened by split of ATP, is used up in pulling them. So this is called as power stroke. Once another ATP comes and binds to it, it will be undergoing a conformational change, because of which it will be leaving the actin. Microfilament को leave करता है, जिसकी वजह से ये पीछे की तरफ चले जाएगा. Okay, so that's called as a relaxation stage. So you have an activation stage, power stroke, and relaxation stroke. So you can see here once again the so-called moment of the myosin conformation made this to undergo so-called cleavage. So once the ATP is hydrolyzed, again this will be binding to it and it will be pulling ahead. Okay, so this way your so-called muscular contraction and relaxation happens. So this is all about your myosin. Coming to another kind of a motor called as kinesin. So what does the kinesin do? Kinesin basically takes part in transportation. So how does it do? It uses microtubule. As you can see here, these all the content that you are seeing here. ये सब बड़ा बड़ा chain जो आपको दिखाई दे रहा है, ये सब जो chains हैं, these are your microtubules. So this is your structure, as I said earlier. This is your light chain, this is your motor chain, and this is your stalk. And the stalk at the end is flexible. Okay, so light chain will be the region for where the cargo is being carried ahead. So ये cargo को carry करते हुए, this should be now moving ahead on the microtubule. So transportation को हम यहाँ पे transport कर रहा है. We have two modes of transportation. One is protein-based transportation, where cytoplasm will be allowing the movement of molecules, and this is one mode of transportation. So look at this mode of transportation. So this is your vesicle secreted from your Golgi apparatus. You can see you have a cargo being transported here. यहाँ पे एक छोटा सा cargo है, यहाँ पे एक cargo है, यहाँ पे एक cargo है. So this way you see all of your microtubules are bearing the transportation. Look at this fellow, he is also dragging something. Look at this fellow, he is also dragging something. So this actually makes a moment of around approximately 80 angstroms. How does it do that? Here also the motor domain breaks the ATP, and when the ATP is broken down, this so-called flexible arms will break all the weaker interactions here, make it undergo certain conformational changes, and this allows this arm to lift and go forward by 80 angstroms. Same way backward, same way. So these two are actually acting like legs, and the motor proteins will be every time consuming energy for such kind of a movement. And that's how your kinesin is going to deliver the so-called goods. Okay, and remember here also the delivery goes from minus n to plus n. Okay, so this is about your kinesin. Now coming to the third category called as dynein. So dynein usually we see हमने cilia flagellum देखे थे बैक्टीरिया में, अभी यूकेरियोटिक flagellum में कैसे होगा देखते हैं. Okay, here you don't have energy generation happening. वहां पे हम देखे थे rotors के वजह से यहाँ पे energy generation हुआ है, लेकिन यहाँ पे rotors है ही नहीं. These are actually linear motors. So how are the linear motors giving you an activity? How are the linear motors able to do an activity? So look at the way this sperm is swimming. Okay, so look at the way of flagella. So the flagella is actually not rotating here, rather it's producing a forward wave and a reverse wave. Okay, so which means that there's a crest and a trough. It's going in a wave fashion. So this is made up of congregation of linear motors called as dyneins. So let's look at the cross section of this. यहाँ पे cut करके देखते हैं. So see, this is how your congregation is going to look like. These are your microtubules, which are in pairs. Now the microtubules are interconnected to a central rod, from where the activity, so-called braking activity, is generated. These all microtubules will be connected to these proteins called as connexins. Okay, and in between the microtubules, you have the wave generators called as dyneins. These are the short linear proteins. So what do? They pull or they make these two undergo certain change whenever a ATP is hydrolyzed. So how do they act basically? Look at here, you have section A, followed by you have section B. So when section B dyneins are active, section A undergoes inhibition. And when section A is active, section B undergoes inhibition, which makes a wave generation happening only on one side, not on both sides. Okay, जब यहाँ पे wave generation हो रहा होगा, यहाँ पे recovery हो रहा होगा, और जब यहाँ पे wave generation हो रहा होगा, यहाँ पे recovery हो रहा होगा. So just have a look at the mechanisms. So now first see, this is your power stroke. So when a power stroke is happening, you can see the binding happening to the lowermost microtubule element, and this makes the microtubule to move, generating a small wave. Okay, so this happens for a short time, and the so-called locking mechanism will get failed after a certain scenario, because you have an inhibitory signal generated at this junction, which makes this not to interact with the lower portion. Okay, so on this entire stretch of microtubule, you don't find the binding. Okay, whereas on the other side, you find these are getting activated. यहाँ पे देख लीजिए ये काम कर रहा है लेकिन ये साइलेंट है. यहाँ पे जब ये काम कर रहा है ये साइलेंट है. Okay, now let's look at how a wave is getting generated from the base. It's coming ahead and it's giving you a bend. Now this is silent. Now this is active. From here again you get a bend. So this way you have alternatively acting dynein portions, making the wave to generate in a forward bend and a reverse bend. Okay, and this is how the swimming is possible in case of our eukaryotic flagellum. So this is basically a linear based motor. Bacteria में rotary है और यहाँ पे linear है, ये भी complex है. Okay, and this is not simply working on a single motor, rather it's working on congregation of different rotors. Okay, so that's how your so-called the cilia or the flagella in the eukaryotic systems function. Okay, so this we are talking about sperm of sea urchin. Our sperms also do undergo the same sort of a swimming. Okay, and most of the eukaryotes also tend to exhibit the same fashion. So you see this is going in a wave fashion. Bacteria में rotary fashion में जा रहा था, यहाँ पे wave fashion में जा रहा है. Okay, so this is all about the molecular motors and the machines. The same thing is being mentioned on your PPT. Okay, I'll share the PPTs with you all. So once this is done, you just try to correlate. Okay, आप आपका किताब से correlate करने की कोशिश कीजिए. So this is about kinesin. So kinesin, I have already discussed this. Movement of arms. So they literally walk on the microtubule from minus n to plus n. And this is the dynein. So usually dynein also have a basal body from where your establishments basically happen. And this is where the energy breakdowns or energy molecules are carried forward. And that's the reason why you see the wave generation comes from this region. So either we undergo a wave pattern leading to generation of vibrations, or leading to generation of what do you say, movement, wave kind of emotion. So that's about molecular motors and machines. किसी को कोई doubt है क्या? Does anyone have any doubts?
एक बात पूछ रहा हूँ सर, लास्ट साल का जो एग्जाम था, मतलब लास्ट जो फर्स्ट ईयर था, उन लोग का सिलेबस और हम लोग का सिलेबस में कोई डिफरेंस है?
है डिफरेंस है. That's what I said, no? उन लोगों का सेमेस्टर पूरा 6 महीने तक चला था, जिसकी वजह से उनका पूरा कंप्लीट सिलेबस है.
हम लोग का कम है.
आपका थोड़ा कम हो जाएगा. अभी यहाँ पे हम बायोरिमेडिएशन पोर्शन कट कर रहा है, एंड नेक्स्ट चैप्टर में इम्यून सिस्टम कट कर रहा है. तो वी हैव अ चेंज इन द कोर्स पैटर्न.
ठीक है सर.
ओके, अभी फिलहाल आप अटेंडेंस दीजिए मुझे. आई विल जस्ट टेक द अटेंडेंस, मार्क द अटेंडेंस फॉर बोथ द डेज. सी आई विल कॉल सर्टेन रोल नंबर्स ओनली दोस रिस्पोंड, क्योंकि इन लोगों का अटेंडेंस थोड़ा कम है. तो जिसको मैं बुला रहा हूँ सिर्फ वही लोग रिस्पॉन्ड कीजिए, बाकी सबके लिए अटेंडेंस लग जाएगा. रोल नंबर 427 आयुष दहिया 427.